I've got a couple of questions to kickstart, but I'm sure that members of the present audience and the online audience have many questions as well. And my first one is perhaps somewhat simple for people like myself, but it's a bit of genetics 101. We've heard phenotype and genotype. Is there somebody that could just explain to our audience the difference between phenotype and genotype?
Yes, sorry, John, we give that a lot of examples. Phenotype is a genetic information you are moving in. Phenotype is the consequences as a it could be some clinical consequences if it's related to a disease, but it's also all the size or hair or color of the eyes and everything. This is the phenotype. And when we talk about disease, the phenotype and also clinical manifestation.
Can maybe simplify this.
You're used to teaching.
Yes. So for the algorithm, it's an essential the algorithm. So if you try, like we did, if you try to make a mouse where there is no algorithm, then both copies of the algorithm are missing. The genotype is both algorithm copies are gone. The phenotype is very, very simple, not today. So that's where it's gone.
Lovely, thank you very much. Now, I'm going to go to the audience and I would like to know, yes, Betty, I'm coming straight over to you.
So I have a question because when Gavin first was a baby, I kept trying, I kept searching, kept asking. And they did, they said there's myelin on his brain. The myelin was not where it should be. And originally that was why they were telling us that he had this hypotonic. So does the myelin and the development of the myelin kind of do this or slows it down, or you know, we never checked it.
Well, maybe without knowing this precisely, but if you think that myelination is like wrapping around myelin around axons and making them, giving them stability, and so it's an important part of the nervous system to have the processes of neurons wrapped around with myelin. And that's cellular stem, and that has something to do with the electricity in the neurons. It's like a cable which is isolated. And making the myelin around this thing is a complicated process where the right amount of myelin is provided at the right time. And the processes of the neurons need to grow at the right time. So if you have any disconnection, so it's important, interesting information if you have any disconnection that the myelin is made at the right time, myelin is protein. And you got the wrong wheels at the wrong position at the wrong time, then you have a problem. So I cannot comment exactly why this would happen, but you can just easily imagine something.
Thank you. And passing to Manu, who has some questions from online.
Yeah, the question of the model, 1801 he asked whether there's any promising without having rocks on the brain or
we are still very far from being able to test any drugs. As we have shown, we have just established the models. We have to identify what's going wrong in these various models, cellular and animal ones, and only then will we be able to test substances to improve this. And it's very hard to say with my work.
Yes.
Right, so maybe I can quickly, because I wanted to say this also in this audience. Maybe very, very simply, we are at a point where we don't even know whether there's a loss of the function or a gain of the function. So there's a protein that's usually has a different function or it lost the function. You can imagine it's a completely different way of treating it. if we gain something new, then we have to think in a completely different way of communicating compared to a loss. If it's lost, we need to reestablish it, right? If it's gain something, we need to delete this. We are at the process, and I think we're getting closer and closer, but we are at the point where we're figuring this out. We have to work it out.
As Rich mentioned, any of that, including like therapies and medications, any of that being collected as well as symptoms, medical history, things like that.
This is really something that we are especially looking at. When some of your children that develop seizures, and each of those kids received the therapy. Some of the kids, the first therapy worked, we would really love to know which one was it. And some, the first therapy didn't work, the second didn't work, the third worked. And it's extremely important for us to know this information. It's also important for us to know the information, which kind of physical therapy we found. I know that, for example, I suggested Antje to go to Cologne for a special program, which is called the Galileo program, which really improved the strength of the muscles. After that, he really was able to learn to walk. And I can, to all the children who have muscle weakness, who have an unsteady gait, I can recommend to go to Cologne, or perhaps there are similar programs in other countries, which really improve the strength of the muscle. I would really also like to learn about the speech therapy. Many of you have had different kinds of speech therapy. Which one of those worked, which one didn't? Where did you see improvement with what? All of this information is something that we actually rely on.
A question from the chat, Raven from USA is asking, for those of us located in other countries, how would you recommend you contribute to your research, example, interview?
So we will ask Nora to send a newsletter and share the content information with me and my colleague, and we'll be having interviews online or Zoom. And we'll also be happy to receive as much medical records as we can.
Yeah, I have also something to add. So I forgot to present it, but we also have a project that is the website where the family can answer some questionnaire, and it's in different languages. So it could also be helpful to the family that don't speak English or they are far away. But it will be really complementary to the register. So even if the family participates in the interview, they can also fill online the questionnaire and we try to combine both information and help us. So I don't have any, but I will pass in the reaction. Maybe you already a message for that because I don't have any flyer. But so yeah, would be also a way to participate.
I was given a question, which I have to read out, because I think that in a way you've answered it already, saying that we're at such an early stage in the research, but nevertheless, do you think that there is a way that other organelle proteins could compensate in some way for a mutated one? Is it something that we can think about replacement therapy? I know this is a very difficult question to answer, but I think it's an interesting one because we look at some of the other rare diseases like lysosomal storage disorders and where we use enzyme replacement therapy. So could we use protein replacement therapy?
Yeah, I think it, again, it's really what I said. If it's a protein that is mutated, acquired a new function, then You cannot replace it with anything else, right? So in this case, it looks like this. And in this particular case, you cannot do a rotational therapy, you just need to get rid of the mutated version. We also would not be, but we will not be able to do any kind of sensation or upregulate any other compensating other proteins. So in this case, not.
Anyone from the audience? I have a question because we have luckily we have you two, all of you, but we have AD1 and 2 on the stage. And we see that there are similarities. Now we have a name for AD1 as syndrome. I'm not sure if we have it for AD1. Question is if it makes sense in the future to join forces and to use the power of a common one syndrome to go out also communication and very clear positioning of work. And in that respect, it comes also to the coding. So the orphan seems coding. Could we, I guess you're asking, could it just be one code? Anybody interested in? I think we've got an orphan, so we see if we have the same answer. So me, I would definitely say yes. Because I don't know what's related to the knowledge of models or what was doing, but yeah, I'm sure we should join the course as a research point of view, as a clinical point of view, and I think you guys are really also an actual point of view. But we'll see with them.
Of course. This is the point, and I think we already discussed that perhaps we should write at some point a review, but we put the reasons why we believe that these are two almost identical syndromes, both in terms of molecular value and clinical symptoms, and we would suggest that I would set something like Argonaut syndromes or Argonaut neurodevelopmental syndrome.
And then we have to ask Nora to which wants to change the name of the application.
I want to remind everyone that there's also an Argonaut 3. The longer we wait, it will be another 3 syndrome. So we want to call it Argonaut syndrome. But yeah. Yeah, it was an Argonaut 4. Argonaut 4 is a little bit different. It seems to be less expressed, more in expression. So there's for sure different, but there's also an Argonaut.
I know that you touched on this, but again, it's a question that was passed to you, and I think it's crucial. It is, what is the first step that we take towards that famous patient registry? How do you want everybody here and online to go about this? Because I've worked with patient registries. I know that they are huge and it takes a huge amount of data and time and manpower to get them up and running, and also to collect the data in a way that is going to be useful to the scientists and down the line, useful to the pharmaceutical companies the day we're lucky enough to find a treatment, because they need the data from those patient registries. So looking to the future and saying this is our biggest next hurdle apart from the scientific side, how do we go about this based on your experience?
So we need a person who is dedicated and full-time doing this, to interviews, analyzing clinical data, and I hope that we have a whole year of discussions in this matter.
I mean, that's the best news I've heard so far, because I always think that patient registry gives power, and that means that through your scientific world, you can reach out to other scientists, other clinicians, and through that we join the dots from the patients that you were showing up on the screen as well as all the others. Now, in terms of diagnosis, what I've heard so far, and forgive me because I'm quite new to all of this, or Argonaut syndrome, is the diagnosis, the time to diagnosis and the cost of diagnosis seems to be two big hurdles for families. In your experience, can you recommend anything that will change the time to diagnosis and possibly the cost to diagnosis?
But I think it's really dependent on the country, so it's difficult to really answer. In France now, we really accelerate some programs, we have programs like genome sequencing, that could be really quite early in the life, once we realize that something was wrong with the people. So really accelerating a lot, and depending on the country, some are really like Netherlands or UK, they're really like far away, and some others are, I don't know in Germany, you guys, like in France, or we are quite, it's okay, but we can still improve, and we are doing improvement, so. And after for the cost, also in France, it's free, yes, only Germany.
I just have to say that there are several countries like the whole African continent where there's absolutely no genetic evidence, parts of Latin America, parts of Asia, and also in the developed countries, not every patient can receive 3 exome sequencing, exome sequencing, they can receive so-called panel gene analysis, which means that only genes that have been previously connected with similar syndromes or similar disease will be analyzed. And as far as I know, there is not a single panel analysis that will be used. So I believe that once it will be, okay, sorry. Sorry.
I would say the more.
But once it's generally included in the panels, based what we know from other diseases that we've been working on, that would be a huge increase of patients.
Great, thank you. So now we've seen children of different ages just at the conference here in the last 2 days. In your experience, and I know this is a difficult question, when you look at a 5-year-old and you look at, say, a 15-year-old, that you've been able to, do you see any changes in the disease? Do you think that the disease progresses? For example, when we talk about Duchenne muscular dystrophy, we say a progressive degenerative disease, and we know what that means. Do we have a picture of this for Arnold syndrome?
So I mean, I have to say that so far I've seen only
Right.
all the information I have, I mean, apart from yesterday.
Yeah.
But from what I know from the colleagues is that there doesn't seem to be a regression. We cannot be sure that there won't be some special cases of specific mutation, but this time point it doesn't seem that there is regression. There is it's going worse.
Yes, good, thank you. That was quite an important question, and I think that as we follow up with the registry over the years, we will get more and more of that information.
That was my question.
Yeah, I think this is very important.
But I also have to say that there are just a few. We don't know many people who have other mutations who are 50 or 60. And the reason is that in most of the countries, even if they are affected, such people get lost. They do not receive the medical attention that they should receive, that they receive until the age of 18. This is a huge problem worldwide.
Yes.
What is the oldest known case of ALT1?
45.
Just for the online audience who might have missed the question, Ashley asked who is the oldest Arnold syndrome patient, and we heard that it's 45-year-old. Are there any other questions from our live audience? Yes.
No, this is not really a question, more of a request and following up on what double. My name is Martin, I work at Bob. And would like to do streaming online library we have in Bob. And so it would be extremely important if the therapies you've done, where drugs have worked on any aspect of the disease, anything, on the epilepsy to whatever, it would be important for us to know because that helps us to set up an asset. Because as was described, we have the genotype is one mutation. And one mutation, the phenotype is a lot of different. There's a lot of different things in the dish. And we don't know whether all of the things you see in the dish in that, what do they correspond in your children. So that connection we don't have. If we know that a drug has worked on one aspect, then we can test in the dish, we will see, that thing has changed, that thing has changed. Now we don't mask so also not only the physical therapy, but any kind of dog that's done any super
Thank you very much. Regarding the patient or the parent approach, one question that's really been bothering me, and we've heard about it, is parents instinctively know that there is something wrong with their child, and they come to their family physician and they say, "I can't put my finger on it, but there is something wrong." And more often than not, particularly in those early years, and particularly if it's a first child, the family physician fobs them off with neurotic mother in the back of their minds. And my question really is, how can we help parents to either be forceful or to push or to fight a little bit like Debbie has had to do for many years, and to make clinicians sit up and listen and not label them as neurotic parents. And in your experience, how would you advise parents to talk to clinicians?
There are two aspects of this problem. This is one aspect. What we also observe, not so rarely, is that actually sometimes it's also the parents who neglect the problem. I just want to point it out. But what we have to do as people who work at the university or teaching, we have to educate next generation of medical doctors. And also educate young medical doctors who specialize in the field of advanced. We are doing.
Yes, thank you.
Actually, the question, the related question, because what I strongly believe is that you have to do your jobs and you go to little parents calling you each day, talk about that.
That's fair.
I would, that's what I'm we have a wide escalation system from family doctor to you basically. So taking you as the last escalation step and taking a family doctor as the first step. Do you think they are not steps in between?
Or maybe we have too many steps in between.
This is the question which is different again from country to country. A lot of politics behind it, and I'm sorry, it goes very far beyond
Yes,
the obvious.
yes.
Sorry.
Yeah. I think what you're saying is that's typical of any disease, a common disease as well. We have to go through the steps and to get to the point that people are seeing the right clinicians. Debbie?
Well, I'm a firm believer in parent support, which Nora and I mean, that's why we're here, right? And not to be afraid to reach out to someone as like myself who is willing to speak up, because I mentioned that I had a doctor actually approach me, maybe he was 7 or 8, that maybe I had Munchausen disease. And
By
that was,
proxy.
yeah,
By proxy.
by proxy, and that was like eye-opening. And you realize, I mean, you hear children that die, or in the United States, that they said the parents were horrible parents when they starved to death, but Gavin would have died had we not stayed on top of, you know, getting the help, yeah, to get a feeding tube. So, you know, that I think parents support and, you know, of course, the Facebook page and all, and this, these things.
Yes, connecting therapies. I see Christoph has a question as well.
Thank you. So as a layman, I see that mutation on the same gene can have such a wide variety of effects. At the same time, I see that the same symptoms can be caused by mutations on many genes, diseases, problems, more. So when in the far future we can think about therapies, do you think it's more promising to think about correcting that mutation on that gene, or is it more about saying the patient has seizures or has weak muscles, so we must learn from what was done about the weak muscles resulting from others? So by correcting the mutation, you mean something like CRISPR-Cas technology, so it's like to take out the mutation of the cells, from the brain cells. This was what you were suggesting. I didn't understand what this, but this is something. So there is a huge problem with this CRISPR-Cas approach because we use this approach in research all the time. We know that when we introduce a mutation or when we delete an amino acid, we almost never do only that. We almost always introduce something else somewhere in the genome. And there is one thing that has been dominating the field of medicine in the last century. It means primum non nocere, which means first no harm. So at this point, playing with this something that shouldn't be done. It's not approved for this reason anywhere in the world. The other question is, can we, and at what time point, even if we improve this CRISPR-Cas technology, if we are sure that there are no so-called off-target effects, this is what I mean, that we also introduce something else to change. Till what time point will it work? Because we know that the human brain is completely formed by the age of 5. So even if we correct the mutation at the age of 10, can we really correct the brain? This is something that we don't know. So, and for this reason, I think that the better option is to try to identify the pathways where we can go and try to correct. This is my opinion. Perhaps the colleagues have.
Yeah, I mean, I agree, especially because so you can think with Ag1 and Ag2, but they are maybe doing the same kind of thing, and so there are some convergent molecular mechanisms affected, but you can also have this for other genes involved in the developmental disorder. So we have genes that are involved in kind of the same molecular process, for instance, mRNA regulation or microRNA regulation. And so if we can find some convergent pathway, maybe we could act on this. So after the gene can eventually but epilepsy could be due to many alterations in neural functioning. So we need to find subgroup when we could find common convergence. Maybe we want to work with this.
Yeah, maybe, I think, putting it in a little more general terms, I think the question was whether one should really work on this Argonaut protein at all, or treat more or less the disease that we experience, right, in a way that we have already drugs against sleeps and things like that. And CRISPR-Cas is one particular thing to work on such a gene, which needs to really, affect our DNA, and this is a little bit problematic. But working on another level, there's other techniques to target the mutated Argonaut proteins on a non-genomic level, on higher levels. I think that is also something one could envision, maybe in a longer terms, because we need to know what exactly happens. But I think when we know what happens and we can early treat the patient very early, we might be able to at least maybe cure some of the parts of the development later. It's not really what's coming later. So I think it's a mixture of both. So we would have to go both.
Thank you very much. A question.
Question from Argentina, where mother, I think, of the PTSD trial wrote, and you already said, we have no advances in terms of accurate diagnosis. And she asked whether you are aware of any APO research in Latin America, especially in Argentina. I think you already said it.
Research.
And I guess the clinicians would be reaching out to you.
Clinician.
Yes.
Take my research.
Oh, the last question.
So the discussion is when we first identify the disease, deal 1 or 2, and you mentioned timeline often. I feel like it's actually happened really quickly. From the time my son first had to be in a room with other people, research something on the OR, I think that it's actually going really quick. You mentioned timeline with for any kind of
I'm not a biochemist, I'm not a great specialist, I can just say what I think about it. So what I find really interesting, that was the last couple of days and also this week. So the studies and the papers that came out like 1.5 years ago, now we know quite a bit already about the mutations. We have this association of the learning. So this is really very far, because we have not seen this before in other agencies. So it's really going very far. I don't think I can say anything about panels, because the complicated thing here would be that there's a lot of different mutations. I think every patient will have to do specific treatment or something. So say, but what at least RNA, and this is what we are working with benefits quite a bit now, is that RNA research and RNA therapeutics is really boosted, first of all, during the COVID mRNA vaccination. So it's really a lot of acceleration in the field of development. And then we have for other rare diseases, we have RNA molecules, for example, extracts, for example, this is approved, and this is really in an industry too, but things are moving into the market, and we also get something out of it, which is always important. And that makes things not much faster as it used to be. Right? When I was a student, now exactly 20 years ago, really SMA is finding much better. Very complicated disease also in terms of gene regulation. It was science fiction. The people kept on going, and at some point there was a drug on the market, actually a completely new class. I think that opens quite a bit now, and I think that accelerates the timeline for see as well. So that's my opinion.
Yes. And to add to that, of course, we can learn from some of the other diseases that have some similarities. I think this is absolutely magnificent. I've been told to wrap it up, so if you have questions, please ask them during coffee.
Sorry, I can't hear you. Now you can run this.
Fantastic, thank you very much. Thank you, that's great. It's not a question, it's a comment. I think we're going to need several $1 million to get this going, and a lot of wishing and positive thinking, but this is a start. Being here all together, learning from the experts, who, as the horse said, they are learning through you as well. So with that, I thank our panelists and I thank the audience, both live and online. Tea will be served shortly. Thank you very much. Hands together for our